by the salinity. The first thing that we must remember is what happens when the two different species are in contact. Normally, there are reproductive barriers and there are no gene flow between them. So when we see the genetic structure of this, we see that the two different species are different in genetic codes. But sometimes this barrier disappears and the both species can reproduce. When this happens, the offspring is called a hybrid. So we can define now what is a hybrid zone. It's when natural hybrid can make occurs among boundary sympatric species. There are different scenarios for the hybrid zone. The first one is called non-stability, and this happens when the hybrids are less fit than the parental. They cannot reproduce, for example, because they are sterile. So when we see the genetic structure, we can see that the hybrid has a half of genetic of one parental and the other half of the other parental. The second scenario is called stability, and is when the hybrid persists and can produce F1 generation, F2 generation, or also reproduce with the parental and making bioprocess. The last scenario is when the productive barriers are waking and the two species become one. This is called a hybrid speciation. Our hybrid zone is in South America, in Argentina, very close to the sea, and is a lagoon called Mar Chiquita. This lagoon has sea connection, but also have a lot of freshwater rivers that flow inside. Mar Chiquita lagoon have a great biodiversity, and is actually a biosphere reserve of UNESCO. Since the lagoon has sea connection and freshwater connection, its salinity can vary a lot, and it depends on the rain, the wind, the tide. Our model for understanding the hybrid zone is a group of fish of the genus Orontestes. This group has 19 species. Some of them are marine, others are freshwater. And for this study, we report 3 different morphological species. One of them freshwater and 2 marine species. However, until now, there are not molecular tools that allow us to resolve the phylogeny of these species. And some papers say that there is a promiscuous speciation among the species of Orontestes genus. And this is because mitochondrial or microsatellite marker cannot resolve the phylogeny. So to be a little more complicated, in Mar Chiquita lagoon, we can found intermediate individuals. These are intermediate because they have some morphological characteristics of Orontestes, the freshwater species, and other morphological characteristics of Argentinensis, one of the marine species. The Orontestes are a very important group in South America because the quality of its flesh, and also it has a long history of introduction, and actually it's a very, very popular fish for sport and for commercial fishing. Our sampling was inside and outside the lagoon with 3 morphological species that live in both inside and outside, and the intermediate morphotype that live only inside Mar Chiquita. The method was RAD sequence. With this data, we perform trees population analysis and some hybridization analysis. The first result that we found is a completed species boundary delimitation in all of the species analyzed. Both individuals inside or outside the lagoon of the same species are grouped. We can note here that Odontestis platensis is a more distant species, and that bonaerensis and argentinensis are sister species. We can look also the intermediate individual between these later species. So to understand deeper what happened in this relationship, we do a population analysis. This result is very informative because first we can see that the Argentinensis, is split in two different populations. One lived in the sea in 4 different localities, and the other lived inside the lagoon. Actually, this population was recorded as resident of the lagoon. They cannot go out the lagoon. In the other side, we can see the blue species, the freshwater species, that are a single population. There are not genetic difference between the population inside or outside the lagoon. So when we look inside the lagoon, we can find 3 different groups. One in red, that is Argentinensis, other in blue, that is bonaerensis, and between them, we found the intermediate individuals. Yeah? And this result, remember, is a case of hybridization. We do also, between these 3 groups, some hybridization analysis. We found again that the intermediate individuals are between these 2 pure species, and they are hybrid. When we do some hybrid index, we found that all intermediate individuals are very close to 0.5 index of hybridization, and this is because all of them were recorded as F1 generation. So now we can discuss 2 different situations. One is when the salinity is high. In this case, the marine species can enter in the lagoon. The other situation is when the salinity is low, and here the freshwater species can enter in the lagoon. So these 3 species can be sympatric. And our result shows us that the 2 marine species actually don't reproduce between them because there are reproductive barriers. But when the marine species with the freshwater species are in contact, they can reproduce, and the offspring are hybrids. So, in conclusion, we report 3 morphological species, and we found that the species boundaries are very clear. We report also some structural population in one of the species that different the brackish and marine population, and also we found that intermediate specimens are hybrid, and they are all F1 generation. So now we can look again the 3 scenarios, the 3 potential outcomes in the hybrid zone. And we can see that Odontestis in the Mar Chiquita lagoon are in a no stability hybrid zone scenarios. So, and we also can suggest that this scenario is constrained by the salinity of the lagoon. So, thank you so much for coming again. Thank you.